supposed to be an evolved species?
Well, I am glad you asked, Jennifer. Let me tell you why. Let me introduce you to the wonderful world of proteins. Alright, proteins are composed of sequences of building blocks called amino acids. Intermolecular forces inside the molecule cause many proteins to bend and fold over themselves, creating three-dimensional globular shapes. The complexity of these structures make proteins the most versatile and diverse macromolecule known. It also means that proteins can have many different functions, including their role as enzymes. Enzymes are a very important part of any reaction because they lower the activation energy needed for the reaction to begin. Without enzymes, important bodily functions might not be able to occur just because there's not enough energy to begin the reaction. Your problem with gout is due to a lack of an enzyme called uricase. This enzyme helps catalyze the breakdown of uric acid crystals that form within your joints. Luckily for you, I happen to know a few experts.
Terrestrial mammals produce insoluble uricase, and for most mammals, uricase still breaks down the uric acid that causes gout. So why not us? Well, we can analyze uricase as a mammalian homology. That is, this trait multiple species likely derive from a common ancestor. And how do we do this? With a tree! Not that kind of tree. We have living species at the tips of what is known as a phylogenetic tree, and they each have alterations of uricase. Well, we've analyzed the genetic code of each of these alterations. And based off of these codes, we've been able to resurrect models of old uricase to mimic what we believe to be versions existent in ancestral species. So we create this visual representation of how uricase has evolved in the past 94.2 million years, between us and ancestral node 19, the common ancestor of all mammals. Basically, we have a trend decreasing enzymatic activity as we travel from the root and to the tips of the branches, with this shaded region after ancestral node 30 representing where uricase has become completely inert. This is where primate/human ancestors completely shut uricase down, creating a pseudogenetic uricase. But why would a change that has a negative implication take place to begin with? It's simple, really. Evolution doesn't create the perfect species. It's a process over time that prioritizes the fitness of a population in a given environment. Lots of temperate forests, lots of fruit, lots of fructose, lots of energy for those who can find a way to use it. More on that later.
So how did we test this? By using transfected human cells with two types of ancient uricases. One test was a co-localization study on the transfected cells. The control of this experiment completely lacks color, so any color is significant. Yellow in the third column shows the places where the uricase and catalase spatially overlap. The level of uricase activity was then calculated by using spectrophotometry to measure the decrease in uric acid. Transfected cells had higher levels of uricase activity, which shows that the uricase was successfully transfected. The results of a Western blot show that the lysates of cells with ancestral uricase had higher levels of AMP kinase, which inhibits the production of triglycerides.
Triglycerides, now we're getting to the root of the problem. Researchers have suggested a relationship between triglycerides and uric acid levels. Lower levels of uric acid in transfected cells compared to control cells as shown in the left of the graph, let AMP kinase levels increase, which in turn inhibits triglycerides as shown in the right of the graph. The relationship supports evolutionary explanations for the decreased function of uricase.
The loss of uricase activity is during a time of global cooling that decreased abundant rainforests to lean temperate forests, forcing humans to adapt and survive. The ability to better convert fructose to fat and stimulate foraging via uric acid became crucial. Yet, in present-day humans, this causes uric acid buildup, leading to gout, which is caused by hyperuricemia, excess uric acid in the blood. Also, the conversion of fructose, encouraged by excess uric acid, has contributed to the rise of obesity and diabetes. This knowledge helps us engineer a solution by using recombinant human uricase.
Wow, isn't science amazing? It's so cool that learning from our past can unlock solutions in the future.